with us every week. We've been doing this for about a year now, and it's been really fun to really kind of take the reins of our health and wellness and really to basically be the CEO of our own health, right? This is what it's all about. Sorry, was there a sound check thing? We have two special guests on today, Dr. Christy Williamson, who's a doctor of clinical nutrition, as well as Michelle Gottfried, who has a master's and is a clinical nutrition specialist. And we're really excited and delighted for them to talk today about nutritional genomics for health and wellness. So thank you so much, Dr. Christy and Michelle, for coming on today.
Thanks for having us.
Yes, we're really excited about this. We're going to do a brief update. This is very brief, but I will just say one thing about COVID and vaccines is that I think we're still in the same stage 1C, 1B, 1C. I know in Maryland, Governor Hogan, I believe, is going to roll back some of the restrictions on Friday. So that is happening. We'll have to see what Montgomery County does, but I know that is what is happening at the state level there in Maryland. I think one comment on the vaccines, and then we'll do more of a Facebook Live on this in the future. I think Jen and I are going to plan this, but we'll do a Facebook Q&A if anyone's interested on more things about COVID and vaccines and the immune system and B cells and T cells. If anyone's interested in that, just put in the chat yes or something, or Facebook or something like that, and then we'll do a Facebook Live about that to discuss that. If no one wants another Facebook Live on COVID, then that's fine too. Okay, yes, one person interested, so I'm definitely going to do that. Great. Oh, I totally didn't want to do that, sorry. Let me just see. So I am going to just talk real briefly about the upcoming schedule. So we had chronic fatigue, talking about that with Dr. Hirsch last week. Dr. Williamson and Dr. Christy and Michelle are here today talking about Nutrigenomics. Next week, we have Dr. Kynes on to talk about functional nutrition for Epstein-Barr virus, and she's written a great book about Epstein-Barr. And then the next Thursday after that, which is the last Thursday in March, we have our friend Peter Liebenthal here from Alexandria is talking about Qigong, and he'll be doing some Qigong and demonstrating that, so that'll be great. So we're going to hand it over now to Dr. Christy Williamson and Michelle. I'm going to stop my share for just a second. So let me see if I can get to their bio. So Dr. Christy, Christy or Chrissy? It says Chrissy, so I gotta make sure I say that right.
I go by either, it's fine.
Okay, all right, so Dr. Christy has over a decade of experience as a leading expert in the field of nutritional genomics and is passionate about exceeding all of your health and wellness expectations. It looks like you're also involved with legislative process and advocacy, which is interesting, and finding creative solutions to complicated problems. Certainly in the functional model that we always deal with that. Likes to find the needle in the haystack, and like that. On weekends, you don't mind me saying that, on weekends, Dr. Christy can be found kayaking or hiking with her two daughters or catching up in the latest nutritional genomics research. I'm kind of like you, I like to do research for fun. That's part of the fun. And Omics is Christy's latest project blending technology and big data. So that's super exciting. Thank you so much, Christy. And Michelle Gottfried is an MS in CNS, a Master of Science and a certified nutrition specialist, and is Omic Diagnostics leading genomics certified nutrition specialist, and has trained with Dr. Christy for the last 2 years. And Michelle is a leader in the field and hopes to develop, curate, and deliver new genomic panels. So we know that in functional medicine, that in a way, nutrigenomics and all the omics that are happening is almost like functional medicine 2.0. This is the future, which is right now. So I'm really excited to talk with both of you. I think we're all excited here to talk about this. I think maybe go ahead and share your screen and we can go from there.
All right, sounds great. You're welcome. Part always takes a second. I don't know why. There we go. All right. Well, I'm happy to be here, guys. Today, we're actually going to learn about looking at metabolomics as a measure of epigenetic expression in clinical practice. So those are a bunch of big fancy words, and we're going to try and what I call take the teeth out of these things. So everybody talks about how it's big and scary, and biochemistry is scary, and nutritional genomics is scary, and all of these things. So we're going to try and defang it and make it not so scary. And what I ask that you do today is maybe unpack some of the things you might have learned, either reading literature or reading a book or learning things from the internet or other places before. Because we, as the thought leaders in the industry, we still argue and debate about a lot of these things, but when you're actually applying it or translating it from the literature and actually putting it into clinical practice, you have to make it usable. So that's what I want you to do today, whether you're a provider, a clinician, or whether you're a patient listening in. It's important to be able to break these things down and put it into pieces, sort of like word blending. You know, when you're learning to read when you're little, you know, when you blend the words together, that's what we're going to do today in terms of clinical practice for nutritional genomics. So trying to make it simple. So here we go. These, of course, are my companies. With Nutritional Genomics Institute is the practice. I'm at Omics, where Michelle primarily works. She does work at the practice as well, but Michelle is she's my master here. She writes all of my genome panels for me. She is a former accountant, so she is a spreadsheet master, which keeps me organized. Thank God for that. And then at SnipEd was where we teach other providers. So we teach other doctors how to do this, okay? So really important to be able to spread this type of functional medicine or systems medicine throughout the world so that we can actually do, as Dr. Wong mentioned, functional medicine 2.0. All right, so let's kind of go back in time. So if you've been looking at nutritional genomics, Jose Ordovez, this definition is his. It's back in 2004. And gosh, guys, it's almost 20 years ago. We can talk about age if we want to or not. We can skip that part. But he basically started out with this amazing definition. And this was before nutritional genomics was even in Wikipedia, but it did cover the two branches. And it said nutrigenomics explores the effect of nutrients on the genome, proteome, metabolome. What are those? And then something called nutrigenetics, and it said the goal is to elucidate the effect of genetic variation on the intersection between diet and disease. And this was the working definition that we had for almost 20 years, guys. And if you are not familiar with nutritional genomics, you still kind of go, what does that mean? What does it actually mean? And it's still really hard to kind of unpack and focus on that. So we're going to start to break that down. And as Dr. Wong mentioned, I work in legislation. I have done some lobbying in my day. So one of the things that I wanted to do was actually define what is nutritional genomics? So I actually created a statutory definition of nutritional genomics in 2016. It's only 5 years ago, guys. So before then, there was no regulation. There was no definition in any legislation anywhere in the country. Okay, so I don't know if you guys know much about politics, but maybe after this year, we've kind of figured out that most politicians don't exactly get along. But to do this, we had to get the American Medical Association, the naturopaths, the dietitians, the nutritionists, basically everybody had to agree. And by the way, my bill passed unanimously, which was a feat in its day. But we got a working definition. We talked about how genetics affects gene function and genetic variation alters nutrient response, and dietary and lifestyle choices influence the function of biochemistry and things like that. So we got it in there. It wasn't perfect, but at least we finally had something in a statute, okay? And this has been replicated in a couple other states around the country, which is pretty cool. The point here, guys, still really new. We went from 20 years ago to 5 years ago, and gosh, okay, are clinicians actually practicing this? Not a whole lot. So what do we do? All right. Well, when I teach, when we work at NGI, we basically need to break this down. What the heck is nutrigenetics and what is nutrigenomics? Simple definition. Nutrigenetics, guys, is disease risk prediction, okay? I want you to start thinking about odds risk ratios. And if you don't know what that is, I want you to think about like BRCA1, okay? If you have BRCA1, you've got an 8 magnitude risk or an 85% chance of getting breast cancer. This is not fixable, okay? This is something that happens when you have this gene mutation. Your doctors may recommend, gosh, you should get a mastectomy. You should consider a hysterectomy. This is not something that's going to change, all right? Is it influenced by nutrition or lifestyle? Sure. There are things called zinc finger genes that are above and below BRCA1. Yeah, it can be influenced, but you cannot actually change the expression of BRCA1, okay? This is not fixable. This is what I want you to think about when you think about nutrigenetic, okay? Genetic, you inherited from mom and dad, okay? Not fixable. Now, when you think about nutrigenomics, I want you to think about anything that you can measure that's going to have an impact on your gene expression, okay? So it's the measurable effect of diet and lifestyle on the genome. So if you can fix it and it has to do with nutrition or it has to do with lifestyle, then this is nutrigenomics, okay? This is always a fixable thing. So it's two really simple, easy definitions. And remember that we as thought leaders still debate about this. The American Nutrition Association just put out a white paper in the last year in 2020. It took years to come up with this white paper on what functional nutrition was or personalized nutrition was, okay? So, and there were dozens of people, myself included, who contributed and thought about this particular paper. So there are people that try and define what this is, and not everybody agrees still to this day what these definitions actually mean. The point here is if you're going to practice as a clinician in terms of nutritional genomics, these are the definitions that are going to help you do it. All right, so a couple of other definitions that I want to help you kind of unpack. An enzyme, all right? Enzymes are often called genes, and this is because genes are enzymes in proteins, okay? This is confusing for a lot of people. Now, when you think of a gene in the body, okay, I want you to think about it being active, okay, especially in terms of nutrigenomics. Now, remember, this is because we are measuring it. So our celebrity SNP, MTHFR, it's actually an enzyme. It's moving throughout a pathway, okay? So when you hear something in nutrigenomics, I want you to think, okay, it's MTHFR, it's a protein, it's a gene name, it can also be a SNP, and it's also an enzyme, okay? So it is moving. So in nutrigenomics, genes equal enzymes, all right? So we're just trying to unpack these things just a little bit to make things a little bit easier in terms of functionality. The same thing happens when we are thinking about cofactors, okay? So cofactors are essentially catalysts, okay? So these things are often vitamins and minerals, so things like vitamin B12 or magnesium or something like that. And sometimes they get consumed by the enzyme or the gene. But what I really want you to think about is that these cofactors are what make the enzymes or the genes go. They make them work, okay? So in nutrigenomics, cofactors equal catalyst. Now, for my true science people, my true chemistry people, I know this can be a little bit challenging because we've learned that these definitions may mean something else. So it's the suspension of disbelief a little bit. But when we actually think about them in this way, and when we're gonna have a little demonstration here in a second, it actually makes it easier to think of them in this function, making them work this way. All right. So here's the little enzyme function review, okay? So especially for my people who this is new for them, I get a lot of questions, especially when you're looking at a pathway, and that's why we're going through this. So I just want you to kind of walk through this with me, all right? So in each time we have a basic chemical reaction, we have a starting substance called a substrate. We have a cofactor or a catalyst that makes it go, and then we have a product, okay? So when we have a down regulation, which is what most SNPs are, or most genes are, okay, they don't work quite as well. Now, I want to make a point here. This is not a point mutation. A point mutation would typically mean that it doesn't work at all, okay? Now, if we have a SNP, like MTHFR, it's still going to work a little bit, even if we're homozygous here or have two bad alleles from both parents, okay? So when this happens, we're essentially going to have low levels of the product, and we're going to have higher levels of the substrate. And typically we are going to have low amounts of our cofactor, okay? So, and the reason that we're going to have low amounts is if we had higher amounts here, then the enzyme would go, because remember the cofactor makes it go, and then we would have more balanced amounts of the product. Now, sometimes we have enzymes that are upregulation. So CBS, or cystothione beta synthase, is a pretty popular one that most people know about. We still have a starting substance, we still have a cofactor, and we still have a product. But in this case, we go way too fast, okay? So we're gonna run through this enzyme. We're gonna use up our catalyst or our cofactor. So in this case, we're gonna have higher amounts of the product on this end. We're gonna have low amounts of the substrate, and we are absolutely gonna gobble up all of that cofactor. So we're gonna have low amounts of those cofactors. Okay, so this is pretty much our basic enzyme review in terms of functionality. And this is really important when we start looking at genes. So each time this would represent a gene or a SNP. Okay, so really, really important to get the basic concepts here. Another basic concept, SNPs do not always equal disease, all right? So people get this in their head. One of the things that always trips people up when we look at like our optimal diet panel is lactase persistence. So it'll show up green on their panel and they're like, oh yay, I get to eat dairy. Well, actually the red or altered allele is what actually means that you actually have lactase persistence. So SNPs can be good, bad, or have no impact at all. And the other thing when we're looking at clinical practices, remember, nutrigenomics means you have to measure the activity, okay? So, gosh, all right, Dr. C, how in the world do we measure SNP activity? With clinically validated biomarkers, all right? So this is not some voodoo stuff. We're not like, ooh, we're gonna measure your SNP activity by, checking out, whatever else is going on. So we're gonna be looking at organic acids, amino acids, stool tests, blood tests, metagenomic testing if you're getting super fancy. So there's all kinds of things that we can do. Now, one of the things that I want to introduce to you, and we're gonna call it a concept theory. This is not because this is not a validated theory, but this is something that we do not have a plethora of data in the literature. And we at NGI are actually working with some of the major institutions, academic institutions around the country and quite frankly around the world to have publications on this to show that metabolomics actually validate the genome. But this process here is how we actually measure organic acids or how organic acids are used. The part that needs to be shown is actually how organic acids are activating or showing that this enzyme here is actually a SNP. And if you think about maple syrup urine disease or something like that, this is not necessarily something that's new. So, but when we have, again, substrate, and then we're looking at a cofactor. So if it was maple syrup urine disease, this would be vitamin B1. We've got a block here. We would be spilling an organic acid into the urine, and then we would have a low amount of this particular product here. Now, what is interesting here and what we do at NGI is that we are looking at both A, B, C, and D. So we're not just looking at the 1-point algorithm, we look at least 4 points and sometimes up to 10, okay? So that's the cool part of these gene-gene interactions and actually showing that in the literature. So that's what's missing in the majority of the literature that you read when you're learning about nutritional genomics or you're learning about SNPs. All right, now I'm going to let Michelle walk you through a validation example.
All right, so some of us will get a gene report and it will have some SNPs on it, and they will say, do this because you have this SNP. That's really not how it works because they're not validating that that SNP is actually active. So at NGI, what we do is we look at something like glutamate or glutamic acid, which is the amino acid. which gets converted into our calming neurotransmitter of GABA. So we can look at the cofactor, which is B6 or P5P, and if that's dumping into the urine, then we know that we're low in B6, and that means that that GAD gene, the glutamic acid decarboxylase, is actually active. We can also look at our levels of GABA. We have no GABA, we know this is also active. So that's a great example. So never look at a gene and say, I need this specific nutrient just because I have the SNP. You need to validate it like this. So, you know, nutrigenomics is measurable. This is an example of that. And it's the most fun part of my job, taking someone's genes and then taking the functional testing, which would be organic acids or amino acids or functional tests, and actually telling the person, this is why you feel the way you do, because look, you actually have low levels of A, B, or C, and that's what's making you feel the way you feel. So metabolomics is actually functional testing. Some of us know this is like a NutriEval, which has organic acids and amino acids. We can use regular labs. Metabolomics is actually the measurable effect. So the things we can look at in a lab that tell us how the gene is working. What else is on there? Yep, there you go, functional test.
Yeah.
So metabolomics, you know, a lot of us get trained in this in school, but there is an entire database that you can go into you can get lost in forever reading about all these different ways you can measure things. It really does, genes give us a blueprint, but these metabolomics tell us the actual biochemistry of an individual person. And so the two together is like having a crystal ball, in my opinion. I love it.
Yeah, it's pretty amazing. Most people, when they work with us, it's we're going to go through amino acids and organic acids as well, you know, sort of an introduction to that. But the point is that it really is a tool in the toolbox, and that genomics really is the last piece of the puzzle. We often will look at the testing first, and the clinical data, and then the genome comes last. And we already know, the point of being a good clinician at this point is to be able to predict the genome. So that's really what good nutritional genomics practitioners should be able to do, is to actually have that crystal ball. And so this human metabolome database here is phenomenal. So if you are, playing around with organic acids or amino acids, do take a look at this human metabolome database. You will learn a lot of things here. It's a really cool site. So the last little definition I want to talk to you guys about is epigenetic inheritance. So people typically are like, hey, I know, what this means, or I think I know what this means. And so I just want to make one last little association here as we wrap up our, concept theory of nutritional genomics and nutrigenomics. So this is supposed to be the study of gene expression versus genetic mutation, or basically what turns on and off the good and bad genes that we have. These are lifestyle genetics. So these are the things that are supposed to be influenced by all the things that we do or don't do, so age, environment, diet, environment, etc., disease states, generational exposures, so the Dutch famine study is one of those famous studies that people always reference here. So there are three coding systems when we look at epigenetics: DNA methylation, histone modification, and the non-coding RNA associated gene silencing. And again, we're getting into some of those fancy, complicated words. The one that I want you to focus on for nutrigenomics is, of course, DNA methylation. And DNA methylation measured, again, by metabolomics. Now, epigenetics essentially equals nutrigenomics in practice, and that's because DNA methylation equals nutrigenomics, the measurable effect on the genome, because it's influenced by all of these things over here that we can measure. Okay, so we've basically broken it down. So epigenetics equals nutrigenomics equals measurable effect on the genome. Okay, now if it's something else, something more static, something more disease associated, it's essentially nutrigenetics and not really something that we can change per se. We can, of course, influence, and we can, of course, give guidance, and there's all kinds of things that we can do for that. But if we actually want to change genetic expression, then we need to be able to measure and we need to be able to alter, okay? So that is really where we stand in terms of nutritional genomics and nutrigenomics. Before we go on, I'm going to open up the board, I guess, for questions really quickly here. Dr. Wong, do you want to moderate that or anything like that? Everybody good with where we are?
I know this is pretty high level. I know we have a lot of patients on here, a lot of community members, and definitely some clinicians too. But in general, I mean, it's basically, just to summarize it, it sounds like there's things that we can't change, right? There's some genes that we can't change, right? And there's some things that we can have the ability to change through epigenetics, through measuring epigenetics, through DNA methylation, and all these different ways to essentially basically modify cofactors, diet and lifestyle, and a lot of other things once you know what you're dealing with, right? That's what it sounds like. If you can measure it, it's kind of like, how does, sorry, I always like to use these, you'll see I like to use it's like funny analogies, but kind of like Usain Bolt, right? He's the fastest human, or was at one point, I think maybe still. So how does he know if he's the fastest, right? He's timing himself, right? 9 point whatever and 100 yards. So how do we know if we're getting better? I mean, we can feel things, but I did a talk for Georgetown just a couple of minutes ago before this, and it was like, it was fairly basic for the grad students there that were students, but one of the things like hypoglycemia, right? Well, fatigue can be caused by hypoglycemia, but fatigue can also be caused by hyperglycemia. You know what I mean? So it's this idea of we need to know and we need to test and not guess about it. I think it's what you're saying.
Yeah, no, that's exactly right. So, I mean, if we can't measure it and you don't feel better, how do we know it's working? So if you start taking a vitamin, for instance, and you have no change, or if you start taking something or change your lifestyle, and in some cases there may be a change or a minor change, it may be changing one part of the pathway, but it doesn't actually have a measurable impact. So if you either don't feel better or we can't actually see something on a lab change, then why are you taking it? How is it impacting you? This is something that we need to actually see and measure and analyze. So, and I think in so many cases for so long, it's just been basically, oh, just take this multivitamin or just take this, random, slew of either medications or what have you, and not really understanding that it's not going to target just the one place in your body that you're going after. It's going to impact every place in your body. Magnesium works in 300 different enzymes in the body. It's not going to target just the one enzyme that you have an impairment in. And so that's the thing when we're looking at all of these different pathways, that, you have to be aware, one, of where all those different enzymes are, but also measuring and watching. And so that's what makes this so complicated. But it is one of those things where, as we learn and watch and measure, we're able to actually have that level of precision, which is just phenomenal.
Yes, and Mustafa, I always can count on you for good questions, either Mustafa or Amber. Can we measure what specific factor, i.e. diet or exercise, contributes to how much change is affected? I guess we're talking about metabolomics and, the testing.
Yes. So, we are able to, there are certain amino acids, there's certain organic acids that we measure specifically, and we're going to kind of walk through some of those things. So in Michelle's example, it was xanthironate was an organic acid. It is associated with vitamin B6, and it can be associated with multiple different things. So, but what we do is we actually look at the genetic impairment of how many genes are actually impaired that require vitamin B6. And then we actually look at an algorithm that's overlaid with gene-gene interactions. So the likelihood is that it comes down typically to 2 or 3 genes, and then we overlay symptoms with that. So when it comes down to it, our accuracy of which gene is actually expressing is pretty close. So it's one of those things when you you have basically 10 to 40% is genetics. The next, basically getting you up to like 85% is you know, environment and diet and lifestyle and that sort of thing. The last 10% is metabolomics. So, you know, you really have to have that breakdown in terms of clinical expertise to get to that point of knowing exactly what gene is expressing itself.
Great. All right. Well, I think let's head to intro to amino acids here. This is exciting.
Got it. All right, so I promise this won't be too scary, guys. We're not going to get into major pathways or anything like that. And then I'm going to immediately stick my foot in my mouth by showing you a pathway. But don't worry, we're not going to go through it. But, you know, I basically want to introduce you to the folate creation pathway or methyl donor creation pathway, which is over here. This is our celebrity SNP, MTHFR. And the goal of this whole pathway here is to make a methyl donor, and we're going to talk about this a little bit later for our clinicians. This one is a methyl donor recycling pathway, which is what we call it. Again, we have easy ways to remember these things, and this is classically called methylation over here. This is a methyl donor disposal pathway. This is also called transsulfuration. And then this is what we call our biopterin pathway or neurotransmitter pathway. But if you'll notice over here, the ones in red are our amino acids, and there's certainly not all of them listed here. There are many, many more. But let's just say we wanted to know the activity of this gene. It's called cystothione beta synthase. It might be a really good idea if we knew what the level of homocysteine and cystathionine was, or maybe cysteine and cysteine, which are the other two amino acids that live over here. And then maybe also if we knew what the level of B6 was, which is again xanthuronate, that's the organic acid associated with this. So if we're trying to figure out what the activity is of a certain gene, protein, enzyme, then it's a really good idea to be able to measure these things. And notice what the relationship is when they're in different pathways, okay? So that's pretty much why we're looking at amino acids, is to understand why and how they can be used as biomarkers, because they play critical roles in our metabolism. We can measure them in blood and urine. They give us really good information. We're going to use them as part of our systems medicine way of thinking. And again, our goal is to be able to predict the SNPs, and the genome should just be for confirmation, okay? The metabolomics data always does the talking. Okay, that's really the goal here. Now, there are two different types of amino acid testing that you should be aware of. There's 24-hour urine and plasma. Now, when we're doing 24-hour urine testing, this is basically for SNPs and micronutrient deficiencies. There are some variations here that you should be aware of. If you have chronic kidney disease, your creatinine levels are going to be too high. And also if you have low BMI, so typically BMI under 19, you're going to have problems here. So your creatinine levels are going to be too low. The amount of amino acids are calculated based on the creatinine levels. So please be very aware of this. Now in plasma, this is going to be our most clinically studied. We've been looking at amino acids levels for, gosh, almost 100 years now. If you have any sort of neurotransmitter imbalances, this is certainly your preferred form. So like the NutriEval FMV would be the way you would go here. Also, caveat here, your taurine levels are likely going to be high. Your erythrocytes store taurine here. So this person is not having a Th1 shift. They're not going to be highly allergic, and they are also not going to be having issues with that CBS gene or CBS upregulation. So please interpret with caution. And if you have a perfect situation where you can do both, you can do both testing. Again, why is all of this important? Amino acids are everywhere. They build everything. Knowing how they interconvert, so my clinicians here, the concept of homeostasis, the body will always try and balance. There's also something called anaplerosis, where we basically steal amino acids and put them into other pathways. Very, very important. I know biochemistry is everybody's favorite, but it's really important to know about your biochemistry. It tells you how the body is behaving in real time. And this, of course, is the essence of precision medicine and allows you to validate the genome. So the pattern is super, super important. And remember, again, this is just a tool in the toolbox. So Organic acids, very similar in function, again, as amino acids. They do work differently. These, of course, spill into the urine. We're not doing a direct measurement here. These have been used for a very, very long time, primarily to look at inborn errors of metabolism, similar to things like maple syrup urine disease. They almost always have a nutrient cofactor, which will basically help resolve the issue. This is available through Genova Diagnostics in Great Plains and others, but we can measure all kinds of things here. So mitochondrial function, dysbiosis, inflammation, toxicant exposure, all kinds of things, and of course, activity of Snips. So we're really looking for that minor variation here. So you may get lucky and find an inborn error of metabolism, lucky, but you are most likely going to find a gray area in terms of how the pathway is functioning. Heterozygosity, meaning that you have one bad SNP, you can be mad at one of your parents and not both of them. And so this is one of those things where you are able to align to optimal function by looking at organic acids testing. So when you're trying to look at your test, either by yourself or with your clinician, it's really important that we look at something called the quintile range. So just like if you were looking at a traditional lab, we want to make sure that we're in that perfect range in the middle, not too low, not too high. We call this the Goldilocks phenomenon. So if you are down here, like over here in this range, this is actually functionally low in the first quintile. Over here, this is what we would call functionally high. Okay, so this also tells us as we're creating these algorithms and working through these processes that we have some slight impairment over here, okay? And that this pathway over here might be a little upregulated. So as a clinician or as a patient, as you're looking at this information, just because it's in the green doesn't necessarily mean it's good, okay? And just because it's not in the red doesn't necessarily mean that it's okay, all right? So this is one of those things, it's a sliding grayscale and something to be paying attention to, because again, we're looking for optimal function. And this patient, we only have one marker in optimal function here, and this person does not have a B6 deficiency. Good for them. All right, so again, the summary here, tool in the toolbox, okay? We're looking at optimal pathway function. We're looking for SNPs. We're looking for those types of things. And we really want to align either to our optimal diet or our optimal biochemical potential by looking at organic acids. Okay, now we're going to take a quick look at methylation because that's always what people want to know about. If you've had any interest in nutritional genomics, people are like, oh, I want to know about this part. So, all right, here we go. Now, methylation, taking the teeth out. It's a carbon and 3 hydrogen, guys. All right, methylation is the process of going from one group with no methyl group to another molecule with a methyl donor. Okay, we're done. That's all you have to know. Happy? All right, just kidding. Okay, all right, so methylation, what it does, everything, okay? It regulates gene expression, biotransformation, removal of toxins. It controls your neurotransmitters. It controls your hormones. It controls your energy production. It controls how well your skin and bones are put together. It does like everything under the sun. So this is why people care about it. This is why people are like, hey, dude, I want to know about methylation. And they try and control methylation. So let's talk about that. Shakespeare, to add or not to add a methyl donor, okay? So let's talk about this part. When you add a methyl donor or you hypermethylate, I literally want you to think about stuffing methyl donors in your mouth, in your nose, in your ears, okay? Stuff the methyl donors, okay? What is this doing? It's silencing you. You can't talk, you can't hear, you can't breathe, nothing, okay? Silences gene expression, nada, okay? Like our cute little cartoon over here, all right? And then when you remove methyl donors, you pull them out, okay? You're pulling the methyl donors, the cotton out of your mouth, out of your nose, out of your ears. All of a sudden, you can express yourself again, all right? So I want you to think, do we think that adding methylated vitamins is a good idea? So if we add methylated vitamins, methylfolate, methylcobalamin, SAMe, betaine, all of these things, it's going to stuff your mouth with methyl donors, okay? It's going to silence your genes. Oh, wait, I have lots of bad genes. Okay, well, what about all your good genes? Are your tumor suppressor genes working today? How about the genes that help you regulate your heart muscle? How about the genes that help you regulate digestion? How about your hair and skin genes? How about those? Are those working for today? Okay, how about your hormone genes? What about all of those? What about all the good genes that are working today? Because I promise you, if you're walking around and breathing and sleeping and like having, relatively normal function, you have more good genes working than bad genes. We have something called global methylation and local methylation. Now, let's just say you have infertility, okay? And you're supposed to take methylated folate, okay? And you take methylated folate and you're targeting your ovaries. You're like, okay, come on, methylfolate, we're gonna work on those ovaries. Now, when you take methylfolate, do you get an injection directly in your ovary? Is that how that works? Or do you take a vitamin? Okay. Now, when I take a vitamin, it has to get swallowed and then it goes through digestion and then it attaches to my folate receptor and then it gets in my bloodstream and it floats around and it touches pretty much everything else. And then maybe this much might get to my ovary. Okay. So it's going to touch all of those other genes and all those other pathways. And it's going to go to all of those other genes. So we need to really think about that, okay? So we call it playing methylation God by taking methylated vitamins, all right? So we really, really, really need to know all the things, all right? My kids always tell Mom, I need to know all the things, okay? We need to know all the things before we even consider giving a methylated vitamin of any kind, okay? Then I get, oh, wait a minute, Dr. Christy, what if I have MTHFR? I know I have to take a methyl donor. I have to, okay? In fact, I got to take methylfolate. So this is what we typically learn. Methylene tetrahydrofolate reductase. Let's say we've got C677T homozygous, 75% loss of function. Does that say 100% loss of function? Remember, we have that SNP activity. It's only partially reduced, okay? So 25% still works, even if you're red, okay? Even if you're looking at your gene report and it says red, it's a big old nasty red over there, still works a little bit, okay? Still makes 25%. The whole point of MTHFR is to make a methyl donor, okay? And we're gonna go through a pretty nasty pathway here in just a second. Sorry, patients. We'll make it easy, I promise. Okay, so, but the cofactors, riboflavin, NAD, which is vitamin B3, and ATP. ATP comes from mitochondria. Mitochondria, they need B12, B1, B2, B3, they need carnitine, CoQ10, and alpha lipoic acid. Did I say folate? Did we say folate anywhere? No, we didn't, okay? All right, so and another thing, about half of the population on the planet has at least one of these variants. You think if this was so bad and that we all have a variant for this, that we'd be living? That we wouldn't all have cancer or something wrong with us, okay? Really, really wrong with us? We'd probably be dead. All right, so let's see here. And it makes a methyl donor. We need methyl donors, right? They're really, really important because they do all those things that we listed, okay? So let's go through this pathway. I promise it won't be too bad. All right. But we're going to start up here with something called dihydrofolate reductase, which is one of my favorite SNPs, by the way. And we're going to start up here. This regulates how your body actually controls folate coming into the pathway. Now, one of the things that it's actually inhibited here, it's inhibited by folic acid. Michelle, can you tell me some of the places we find folic acid?
Well, in processed food, which most of us are eating, grains and cereals and breads. And so when you're eating those things and you're eating folic acid, if you have a SNP there, and a lot of people do, then you're basically blocking the folate from moving into this pathway at all. So there you go.
Yeah. And other places we find it are vitamins, like junky vitamins. Yeah. So if you're eating cereal or grains or, any kind of fortified food, or you're taking any kind of over-the-counter vitamin, most of them are going to have folic acid in it. Okay. Now, our starting folate is called tetrahydrofolate. And for those of you who use organic acids testing, tetrahydrofolate is the marker for form-aminoglutamate or FIGLU. Okay, so if you're looking at that, so my clinicians that are taking a listen here, if you have a deficiency in tetrahydrofolate, or if you're low here, then your FIGLU levels will elevate, okay? So this is our starting form of folate, and it's actually the usable form of folate in the body, which I know may be a shocker. Most people think that methylated folate is the usable form in the body, but this is actually not true. So we're starting here at tetrahydrofolate, okay? And we are basically gonna skip over here to MTHFR. So MTHFR again has our cofactors of B2 and B3, and its whole point is to make methylfolate, all right? Now, what is the chemical difference between methylfolate and tetrahydrofolate? So methyl tetrahydrofolate and tetrahydrofolate, the difference is a methyl donor, okay? So it has to get rid of the methyl donor somehow to get back up here to tetrahydrofolate. Well, how does it do that? Well, it looks like this superstar over here must be doing something with it, okay? So this superstar is called methionine synthase and methionine synthase reductase. But let's just think about this one as a baseball glove, okay? So we're gonna think about methylfolate as a ball, and this one is a baseball glove. Now, if you have a broken methionine synthase or a SNP there, think about the baseball glove being open, okay? And if the methyl donor comes, if methylfolate tries to come in, it's not gonna catch it, okay? It's gonna bounce off, all right? But if you have B12, imagine putting a sticky coating on there. All of a sudden, it'll still catch it even if it's broken, okay? So, and of course it works, it's a nice round glove and it catches the ball anyway, all right? So the point here is that you're gonna catch the methyl donor here at methionine synthase, and then it's gonna turn back into tetrahydrofolate. So this is the methyl donor creation system. And then when it catches the methyl donor here with the help of vitamin B12, which is the most important nutrient in this entire pathway, the most important nutrient in methylation is B12. So once it catches it here, then the methyl donor actually travels around to methionine and then to homocysteine. And the only difference between methionine and homocysteine is a methyl donor. Okay, so this is what we call the methyl donor recycling center. All right, so this is why it's super important that we make sure that we actually have enough B12 when we're looking at methylation, and we start there first, and we don't actually drive vitamin B2 first, okay, even though that's the cofactor. So this again is one of those things we actually have to look downstream and see what's happening below or next in the pathway, okay? So this is something that's kind of confusing. It takes a lot more thought process to think about in terms of methylation, but super, super important, okay? All right, wasn't too bad, I hope, in terms of pathways. All right, so before we get going on that, any quick questions?
Yes, I have a quick question, Chrissy. Curious your thoughts on using serum B12 or other tests like MMA. What levels are you looking for the serum B12? Because we know that at least the US range is obviously suboptimal. That's not what we would use. But what are just curious your guys' thoughts on that?
Sure. So yeah, so anything under really like 650 or 700 in terms of serum B12 is deficiency.
Wow. Yeah.
Yeah. And then, you know, if we're looking at, so if FIGLU is elevated, if MMA is elevated and serum folate is elevated, you're essentially folate trapping. And so didn't totally get into that just because it was
going to the red blood cell, you're saying.
Yeah, it's, well, so this, if you were to add additional folate into this pathway, so if you were to add in methylfolate here and have a B12 deficiency, then you're essentially going to trap the methylfolate in this position.
It's like the keystone to that, essentially. Got it.
Exactly.
Okay.
Exactly. So your tetrahydrofolate levels or your FIGLU levels will rise, showing that you have a deficiency in tetrahydrofolate. So, and that can happen if you have a SNP in DHFR or if you're folate trapping here. Now there are a couple other caveats with that. Malate will elevate sometimes if you have B12 deficiency because inhibition of the malate transporter. There are a couple of other things that can occur as well, but almost always if you are supplementing with methylated folate and you have Snips here at methionine synthase or DHFR, you're going to have an elevation in FIGLU. But the serum levels, you know, when you're measuring serum B12, the majority of the type is actually methylcobalamin. So know that that's what you're actually measuring. And then when you're measuring MMA, the majority of that is actually adenylcobalamin. So you're actually measuring two different things. Yeah. And so that's something to note as well. And then for folate, it is actually folate is actually methylfolate as well. Okay. For serum, they're both the methylated forms. Sorry, I had to file that really quickly, make sure I was saying that the right way. But yes, for serum folate is actually for methylated folate as well. But it is one of those things where, you know, we're not actually measuring tetrahydrofolate in serum, which is actually the metabolite that you need to be looking at to see if you're folate trapping.
Got it. Thank you.
Mm-hmm. All right, so what are the real consequences of having MTHFR, guys? So, because everybody comes in, they're pretty stressed out about it, okay? It's like if you are just stepping into nutritional genomics and you're gonna look and you're gonna Google, you're gonna wanna know, oh my gosh, do I have this variant, okay? So the C677T variant may increase homocysteine, but it is not due to the variant itself. It's due to the interaction with methionine synthase. So it's due to the interaction with a baseball glove, okay? So this is something that can be due to genetics. It can be due to environmental issues. So you could have a B12 deficiency due to the MTR issue. It could be another B12 deficiency issue like a transporter issue, gastric intrinsic factor issue. You could be taking Prilosec. You could have pernicious anemia. You could be a vegetarian. There are any number of reasons that you could have B12 deficiency. So it's really important for you to figure out why you have B12 deficiency because homocysteine is actually regulated by three genes. So the methionine synthase or B12 deficiency, zinc deficiency, which is regulated by BHMT, the one that goes through the middle, and then vitamin B6 deficiency, which is the one that gets rid of homocysteine. So the C677T variant, when you're actually controlling for zinc, B6, and B12, has absolutely no impact on homocysteine. Okay, so that is one of the things the literature is not right. So the literature doesn't control for gene-gene interactions. It also doesn't control for nutrient status. So this is one of the things, again, that we're working on trying to correct the literature, trying to correct for metabolomics data. So it's very confusing. We're kind of in the Wild West. We do not have the research that's out there to kind of prove, hey, you know, we have study after study after study of looking at gene-gene interactions, but we know in clinical practice that this is the way that the pathway works. This is the way that the gene works. So it is one of those things where, you know, it can be a little frustrating. All right, and the other thing is that alterations do change methylation capacity. So our primary way of getting rid of methyl donors is through an enzyme called COMT or catechol-O-methyltransferase, which at NGI is our favorite gene. You're not allowed to work at NGI unless you have issues with COMT. And so we're team magnesium and team B12 over there. So we're going to talk a little bit more about COMT when we talk about stress. But you know, we'll talk about that in just a hot second. But if you have issues with breaking down adrenaline, or you have issues like when you take a methyl donor, if you've taken methyl B12 and you get anxious, you may have issues with COMT. So we'll talk about that in just a second. I just want to reiterate here, the whole point of MTHFR is to make a methyl donor. It's really not that big of a deal. I want you to be measuring methylmalonic acid and glutaric acid. Glutaric acid measures vitamin B2. If you have elevations in those things, you basically have deficiencies. So that's really the big take home here. Please, please do not stress out too much if you have MTHFR. So let's talk a little bit about that stress, okay? And then we'll wrap it up for today. So we've all had a fabulous 2020, I'm sure. So norepinephrine converting to epinephrine is controlled by a gene called phenylethylamine in methyltransferase, okay? So there's that methyl word again. And any time that an enzyme is a methyltransferase, it requires SAMe or a universal methyl donor, okay? So essentially, anytime we have stress, anytime we make epinephrine or adrenaline, we are draining methyl donors, okay? So when we drain methyl donors, we take the cotton out of our mouth and out of our ears. What do? We are turning on our genes, okay? Now, we're all born with bad genes. I'm born with bad genes. You're born with bad genes, okay? But it is our methylation status that determines whether or not those bad genes get to yap their mouths, okay? So this is literally the connection between stress and disease in one SNP, okay? And one enzyme, one genetic variation, okay? So the chemical difference between epinephrine and norepinephrine is literally a methyl donor, all right? The enzymatic process that's occurring here, we're basically taking SAMe, or a universal methyl donor, and we're converting it to homocysteine. Now, hold on a second. Homocysteine is a marker for cardiovascular disease. Okay, so not only are we turning on our bad genes, but we're also increasing our risk for heart disease. So when we think about stress and heart disease and cancer and Alzheimer's and all of the other predisposed risk factors that we have, golly, what on earth are we going to do? Okay, so I've already told you how we control for homocysteine, right? We make sure our B12 status is good, we make sure our zinc status is good, and we make sure our B6 status is good. Okay, so now I don't want you running out and just taking those things, because remember that CBS gene might be an upregulation. You should probably test for that. Okay, but zinc and B12, adenosyl cobalamin is your safest. Hydroxycobalamin, by the way, can increase blood pressure in some people, so be careful there. But adenosyl cobalamin and zinc is generally a safe recommendation to control homocysteine levels. Now, the other thing we're going to talk about again is COMT, okay? So this next slide here, this is our phenylalanine pathway. Again, you don't have to memorize this pathway, you don't have to know all about it, but when we are breaking down things like adrenaline, so epinephrine and norepinephrine, these are our chemical names for adrenaline, but it is primarily regulated by this gene, this catechol-O methyltransferase. Its cofactor is magnesium. This is why we take magnesium for sleep. This is why we take magnesium for anxiety. This is why it works so well. Now, for those clinicians in here, vanamandylate, homovanillate, norepinephrine, L-dopa, tyrosine, phenylalanine, thyroid levels, iron levels, all of these things. You should be measuring these things. You should be looking at this pathway very closely to see whether or not these things are off, what the balance is, okay? So you should also be looking at monoamine oxidase. This could be an upregulation. It does require vitamin B2, so glutaric acid as well. Okay, so these are all things that we can look at and measure and determine what the activity of these genes are. But the point is, all of this is about balance, okay? This applies to life and, of course, biochemistry. All right, so that was it for today. I'm going to go ahead and stop the share and open the board for questions.
Well, Christy and Michelle, this is epic. This is the future of medicine and nutrition right here. You've all heard it live here, so thank you guys so much. And I know we have a few questions. Actually, Anina's another functional nutritionist, and she's on here too. Hey, Anina. So she wrote, what testing companies do you recommend for genetic SNPs?
So we typically use either 23andMe or MyHeritage DNA. Unfortunately, ancestry DNA is not CLIA certified. So we typically do not recommend ancestry.
Okay, great. And then this is my question. Do you guys use the NutriVal, or what other things you would use for organic acids and everything? And
Yeah,
amino?
we typically use the NutriEval with the current issues with FedEx. We've been using the Metabolomics a little bit more recently, but that's also a Genova test. But typically we do use
Metabolomics.
Genova.
Okay, I don't think I've used one. To take a look at that. Great. And then and you like the NutriVal more than the organics tests that they also have?
We do use fatty acids in our algorithms. So
Yeah,
yeah.
that makes sense. Okay. And Nina had a question. Do you have recommended doses of B vitamins for both homozygous and heterozygous MTHFR patients? I'm assuming Anita's question means there's, obviously A1298C versus C677T, but it looks like what you said there is that if certain B vitamins like B2, B3, and B12 are adequate, that functionally you would have generally good methylation, or you could have good methylation with optimization of those.
So it really depends on their TCN1 and TCN2 status, as well as their gastric intrinsic factor status. So typically we would be looking at, if they have, multiple Snips in TCN1, somewhere between 2,000 and 4,000 micrograms of hydroxycobalamin. If they have, depending on how many Snips they have in TCN2, same thing for adenosylcobalamin. If there's a blend, then we'll recommend a blend of hydroxy and adenosylcobalamin. So it really depends on their transporter status, depending on what, we would look at in terms of that. So, TCN1 operates in the mouth in terms of salivary absorption. TCN2 is in the gut. So, it just depends on what their actual optimal absorption is.
Great. And when would you recheck, organic acids or amino acids, let's say, after repleting them? When would you recheck a NutriVal and things?
So typically with new patients, we do it every 3 months. So, that's going to be for the first, 9 months, maybe 6 to 9 months. And then after they've reached stabilization, we typically do once a year.
Great. I know we're about over time here. So thank you for staying a little over. Where can people reach you if they would like to find out more information? I think we should put that on there. Maybe Jen could type it on there. But
sure.
Yeah.
It's nutritionalgenomicsinstitute.com.
Nutritional Genomics NGI. Okay, nutritionalgenomicsinstitute.com. Thank you. One last thing I would say about, just the book that came out a couple of years ago by Ben Lynch on dirty genes. I really like that, his point about epigenetics and lifestyle. What is your feelings, thoughts about things like, sleep? I mean, I think you presented that on the, the norepinephrine to epinephrine. That was nice. Stress, sleep, nutrition. What are all these things in terms of lifestyle and nutrition? How do they affect the expression of genetic? I guess that's where nutrigenomics comes in. But I think what people, maybe just to make that point, just hammer it home, since it's so important, is that it's the whole idea of genes are not your destiny, and you can influence this through nutrigenomics, essentially.
Yeah, absolutely. Yeah, absolutely. I mean, lifestyle has so much to do with gene expression. And, I think again, the biggest thing for us is making sure that we, are measuring that, with testing and that sort of thing. So, not only does the patient feel better, but we're actually tracking that and measuring, their progress. But, I think the biggest thing is, working on longevity, making sure that they're incremental changes as well. Nothing really should be unattainable or unsustainable for patients.
Right.
I think that's really, the biggest scary thing. People think, oh my gosh, I've got to go climb Mount Kilimanjaro tomorrow. And it's not really like that. It's really that incremental, gradual change that really makes the biggest difference for most people.
And then one thing I'm curious about, just in general, is, how there's a lot of different diet plans right out there and things like, how do you use nutrigenomics to personalize the diet plan or the nutrition, if that is something that
I'm gonna let Michelle answer that. We do that every day.
Yeah. Okay.
Well, our best-selling panel is called Optimal Diet DX, and we've taken, I mean, I've combed the literature, and Chrissy, for a long time finding SNPs that have to do with how you should be eating. So food chemicals, your nutrient status,
That's awesome.
and we look at the genes that predict how you should eat, how you process fat, if you're a bitter taster, if you should be doing caffeine, lactose, gluten intolerance. So there definitely is an optimal diet for everybody.
Got it. And that's amazing. So you take so the patient would get like someone would get, say I want to check mine. I would get a 23andMe, which I have, I got to dig it up somewhere. I think it was before they made changes that didn't make it as effective.
Good. That's a good
That's
chip.
a better
So
chip.
maybe I'll chat with you about it and we can talk about it.
Yeah.
23andMe or my heritage DNA, and then they run it through your proprietary panels on the SNPs to figure out where I got it. Okay, this is great. All right, well, yeah, definitely check out their site, Dr. Christy's site, Michelle's site, nutritionalgenomicsinstitute.com. Definitely be chatting with you more about my panel so I can take a look. I've been like tinkering around with things, so it's kind of nice. And then how do you marry this, or how do you pair this with like a functional stool test or those type of things? Or even like, do you do intracellular nutritional tests? Just curious if any of that, like some membrane stuff, would make a difference.
Yeah, so I mean, we use pretty much all functional testing. So I mean, like on our GastroDx test, we use a lot of the stool testing things. I mean, people that have Crohn's and ulcerative colitis or just IBS, that sort of thing. So there's certain, like we look at Q2 status and other things on the optimal diet test or the panel, but it just depends on really what we're looking at.
I mean, do you have a patient like also do a functional stool test along with it, or things like that, or is that something that they would do on their own and then
patient is coming through as a they're coming through as a patient and then they're doing testing and then they're doing panels on top of that, then yes, a lot of times they'll do complete hormones testing, they'll do stool testing, that sort of thing as well. But each test has their own algorithm. And so, that's one of those things, depending on which panel you're doing determines what functional test you do.
I'll talk to you more about it. That sounds good. Well, thank you both so much. It's been a pleasure having you on. Learned so much today, and appreciate you coming on. And definitely would love to have you on again sometime. But in the meantime, this is our preview of Functional Medicine 2.0 here. We'll be back next week with Dr. Kynes, who I think you probably know Dr. Kynes, right? Talking about Epstein-Barr next week too, and I'm sure Nutrigenomics plays a big role with that. So I'd love to hear her thoughts about that too. Yeah, thank you so much for the comments. Jim says amazing. Thank you for all the new information. Mustafa did ask, I don't know if you want to handle this one or not. What are the insurance implications? I don't know. Most functional tests, a lot of them are not covered by insurance probably, but.
So if a medical doctor, so if you were to order the Genova test or something like that, a lot of times they will actually cover them. They typically do not cover them when I order them.
We do, I think to answer Jennifer's, I think Jennifer had a question too about that. We do the NutriVal here. I'll probably start using it more now after today, but we do a lot of stool tests here, Dutch tests, things like that. So yeah, so it's really good to kind of weave all these things and synthesize it together. I'm really excited for what the future holds and learning more about all this. So yeah, thank you again so much, and we will talk to you both soon.
Awesome. Thanks for having us.
Thank you.
All right, bye guys.
Bye.
Bye.